Good evening, everyone. Welcome to the Friends of San Pedro Valley Park's Natural Sciences Lecture Series. I'm Mila Stroganoff, Programs Director and host for this evening's webinar. I'm certain that you're just tickled pink that we finally have some decent weather. There's more sunshine and it's warmer, and I'm sure a lot of you have been at the beach or outside and hiking. And I'm so glad that you decided to join us this evening. So, I'm pleased to say that Dr. Patrick Robinson is joining us tonight and will introduce us to the latest research that has been done on elephant seals. This lecture will prove to be a fascinating one. I will introduce our speaker in just one moment. And first, let me just give you a quick rundown of the last 3 months. Working backwards from March of the lectures that we have had, so that it will be a bit of a reminder, and also we'll let you know that we have these lectures recorded on our website, the Friends of San Pedro Valley Park.org. And for instance, in March, if you were with us, you might have heard that we had Dr. Parker Shames and Tally Caspi joining us from the Presidio Trust on coyote research management and coexistence in the Bay Area. In February, we had Dr. Tom Parker, who spoke about how much does chaparral differ from place to place in California, which proved to be a fascinating lecture. In January, Tanya Baxter, another botanist, spoke to us about the flora of the Northern Sierra, botanical encounters of the off the beaten path. So those were the three that we had, and we're going into a different discussion today with elephant seals. But if you have joined us, and if you are going to be joining us or wish to check our website, we have many more lectures that have been recorded, and that should prove to be very fascinating to you if you wish to check them out. And this lecture, I'm very pleased to say, is being recorded and will be posted to the website in a couple of days. So please have a look and tell your acquaintances and friends and so on and so forth that they can definitely tune in, have a look, and listen to the recordings. And I wish to thank everyone who has been with us for these many years and viewing us through Zoom, and for your attendance, your questions, and your comments. Now, I must remind you, our chat is sort of funny. It's not quite the most dependable thing in the world. So if you have any issues, if you have comments, questions, put them in the Q&A section, and we'll deal with them at the end of the lecture when Dr. Robinson will take them. So thank you again for joining us tonight and through all these many, many months and many years that we've been doing this in this format. So recording is going. And second, I'm going to give you a heads up about the upcoming lecture, which I usually do. I'm very pleased to say that John Kipping will be joining us on Saturday, May the 3rd at 7 p.m. He's a naturalist whose lecture is entitled, "There is a Whale in Your Backyard." He will discuss the migration of California gray whales and their calving lagoons in Baja California to summer feeding grounds in the Bering Sea. So this should prove to be a very interesting lecture, and I hope you'll mark your calendars and join us. This is May. So tonight's lecture will follow the same pattern, as I said, with the Q&A at the end. And so questions, comments into the Q&A section. Avoid chat. And now to our speaker. I'm very proud and pleased to say that Dr. Patrick Robinson is with us. He has completed his PhD and postdoctoral research at UC Santa Cruz in marine biology. His focus was studying the at-sea behavior of elephant seals using biologging devices. Specifically, he identified methods for identifying foraging behavior and characterized the extreme accuracy of the migratory movements. In 2012, Patrick was hired as the director of Año Nuevo Reserve, where he facilitates a diverse array of research, teaching, and outreach activities. He has co-authored over 80 peer-reviewed journal articles on species around the world. So let's all welcome Dr. Patrick Robinson. Take it away, Patrick. It's all yours.
Great. Thank you so much, Mila, and thank you for everyone for joining me today to learn about elephant seals. I always enjoy these. Uh, yeah, I always enjoy these talks because I love talking about elephant seals. They're very fascinating creatures.
Can you
So,
increase the volume a bit?
oh, sorry, I can get a little bit closer. Is that better?
Yes. Okay, I'm going to go on
Great.
audio mute and
Okay, great. So I'm going to go ahead and start sharing my screen here. Great. Hopefully everyone can see the screen there. Um, so for today, I'm going to first start off with a little summary of Año Nuevo Reserve and the natural reserve system, and then discuss a little bit about elephant seal basics, just so we're all on the same page about elephant seal natural history, and then do a little deep dive into some current research and some of the cool findings that are coming out of UC Santa Cruz right now. And then we'll leave lots of time at the end for questions, because usually folks have a lot of questions about all of the interesting work that's going on. Okay, so to start off with, I usually just like to show a quick video of Año Nuevo Reserve. It's just an absolutely amazingly beautiful place. I feel super lucky to work there. So if you haven't been out there, I strongly encourage you to do that. Throughout the first 6 months of the year, there's always a bunch of elephant seals out there. So like now through the beginning of June is a great time to visit. Walk out along this public trail here and end up at some public overlooks where there are in this time of year weaning seals and juveniles and eventually adult females.
Patrick, you're breaking up.
Oh, okay.
You're breaking up. I don't know why.
Maybe it was the video causing that. Is this any better now? I'll trust that you guys can hear me. So Año Nuevo is obviously a state park, and that's a really important thing because it protects the land and allows public access. But it's also one of the University of California natural reserves, which is a really amazing designation because this is a network of different sites that are either owned or co-managed by the University of California. And this is Año Nuevo is one of these sites. The university does not own the land because it's a state park, but we have an agreement with the state park to operate the reserve within there. And so basically, it's my role to help facilitate all of the research and teaching activities at the university level. And so it's a great opportunity for me because I get to participate in lots of cool research out there and then teach students and teach members of the community about that. So it's a great time. And one of the things that we do, and we're trying to do more of, is involve undergraduate students in all of the work that we do out there. So we teach a couple of classes where we bring students out on a regular basis. We have this new research ambassadors program that allows students to get involved at a young age, and then they can go off and do many exciting things after that. And yeah, we really think that that has a really big impact on them. This is an amazing site because it's very rare to have a seal colony that is so close to a research university. So we take full advantage of that. And I would be remiss if I didn't mention the docent program at Año Nuevo. It's an amazing program. I think there are about 180 docents. And so my role is to help share the knowledge gained by the research activities, and then they help disseminate that information to the 100,000 plus visitors that we have to the park every year. So just an amazing relationship, and I think we impact a lot of people by doing that. Okay, so jumping into the elephant seal research program, I'd like to start off by talking about the two principal investigators of the program. So this is Professor Roxanne Beltran, who also happens to be my wife, and then Professor Dan Costa. So these professors co-lead the elephant seal research out of UC Santa Cruz. And so basically everything that I'm going to talk about tonight is because of them. So they're the ones that get the research grants and come up with the exciting ideas to go out and do this work. And it's my job to help facilitate that and help collect data and those sorts of things. So just like to give them credit. Okay, so elephant seals. So a lot of folks are very excited about elephant seals because of what they see during the breeding season. These big males will, oops, these big males will engage in some pretty gnarly fights to maintain their dominance hierarchies and to have breeding access to the females. And those females, exist in harems during the breeding season and give birth to a single pup, which they nurse for about 1 month. So here's a cute little pup that's just taking a little drink. And then within that month, that pup has to go from about 35 kilograms all the way up to about 120 kilograms. So they kind of balloon up into these little blubber balls and then venture out into the world. And that's our opportunity after their mother leaves to give them a flipper tag and start studying them. So we have a pretty big effort each year to put out little flipper tags in the rear flippers of the seals. So each of them is uniquely coded with a number or letter sequence, and all of them are green. Each elephant seal colony has its own unique color, so we can instantly identify an Año Nuevo seal as opposed to a Piedras Blancas seal or a Channel Island seal. And so each of our seals, about 500 per year, get one of these flipper tags, and about 200 of those weanlings each year. We also weigh and measure. And that's the whole basis for our entire demographic research program. And I think it's safe to say that the elephant seal demographic research program at Año Nuevo is one of the longest running marine mammal demographic programs on the planet. It was started by Dr. Bernie LeBoeuf back in the late 60s and early 70s, and it's been going ever since. And that's just a really unique thing, and especially in today's funding cycles that are very short, to keep a very long-term research program going. So we spent a lot of time going out there during this time of year, putting out those flipper tags in the young animals, and then also looking for those flipper tags throughout the rest of the year. So we often see a group of seals like this during the molt, and you can imagine it's a little bit difficult to find those flipper tags in that sea of animals. So we have a couple of tricks up our sleeves in order to collect the data that we need to collect. One of them is, of course, using binoculars and cameras to get a zoom in of those flipper tags. So we do spend a lot of time just walking around the colony with cameras and binoculars, trying to read those flipper tags. And once we do, we can do something that's pretty clever, use Clairol hair dye to put a mark on the seal. And that is completely non-invasive and doesn't cause any damage to the seal. It just dyes their fur. But that allows us to identify that seal until the next annual molt, so up to a year. So what we can do is walk around, find a seal that has a flipper tag, then we write out that same number that's on the flipper tag on this little stamping tool. Very high tech, it's just a wooden stick with a little plank on it. And then we stamp that number on the side of the seal. It dyes the fur. And then you can see we have a nice match there. So the flipper tag in the lower right there is 9961, and we have the mark 9961, and we enter that into our database. And now those flipper tags and that dye mark is linked. And then whenever we see this animal in the future, we know it's that same animal. And that's the basis for a long-term mark recapture study. And what's really neat is that the animals have high site fidelity, so they will come back very reliably to Año Nuevo throughout their life. We do have a small amount of immigration and emigration that happens, but most of the seals that are born at Año Nuevo will return to Año Nuevo. And then we end up with a nice history like this. So this is representing just one animal. And you can see that each row corresponds to one observation of a seal. And they're kind of broken up into blocks, which represent years, so 2007, 2008, 2009, and on. And you can see we have many observations of each seal each year. It usually gets a dye mark added, and then we observe that dye mark. And then we do all sorts of interesting additional observations, like whether it has a satellite tag or time depth recorder, if it has a pup, its molt status, if it has a shark bite, any other interesting observations that we have. And that's an interesting thing to do for an individual, but it becomes even more interesting once we accumulate that information across hundreds, or in the case of this study, thousands of individuals across decades. So we can answer some pretty big questions. So one of the questions we can answer with this mark recapture study is, how important is a mother's experience with raising a pup? And we can take decades of data, it takes a very long time, but we can address that question. And it turns out that as a mother gets older, she is able to produce a larger pup at weaning. So she's able to give more resources to her pup, and that pup has a higher likelihood of survival because it has more fat on board at weaning. It can survive for a longer time, and that's an important period for that pup because it's learning how to swim and forage and make a living on its own. So we can see here that if a mother gives birth at age 3, the average size of her pup is less than of her weanling is a little bit less than 100 kilograms. And there is not a very high likelihood that that individual will survive that first year of life. But quickly that increases, and by age 6 or 7, that's when we identify the animals as sort of prime age. That is elevated up to about 130 or 135 kilograms. And that little bit of additional mass goes a long way with helping them survive that first year of life. So really, it's the mother's experience and age that helps her produce a pup that's more likely to survive. And then we can ask a different question. How likely is a certain individual to give birth to 0 pups or 1 pup or 2 pups or 3 pups in her lifetime? So, in humans, obviously, most of us, the females in our population, might have 2 kids on average, and that's about enough to keep the population going. For elephant seals, it is very skewed. So if we look at the black lines here, you can see that most female elephant seals that are born this year will not survive long enough to give birth to any pups. So this is the lifetime reproductive success of, 83% of females is 0. So, if you're born at Año Nuevo this year as a female pup, you only have a 1 in 5 chance, or a little bit less, of having a pup in your lifetime. But if you do survive to adulthood, then you have the chance of living a long time and producing lots of pups. So we do have some examples of females that we've deemed super moms. They can have upwards of 15 or 16 pups in their lifetime and have a very disproportionate effect on the next generation, which is pretty exciting. Okay, so that's sort of some of the on-land stuff that we can learn. One of the things that we're very interested in is what the animals are doing out at sea. So they're spending most of their time, most of their life is out at sea, hundreds of meters below the surface of the ocean. Very difficult to study them out there. And that's why Dr. Bernie LeBoeuf, when he first started studying elephant seals, he studied the on-land behavior because it's really difficult to study what they do out in the ocean. So one of the major drivers of beginning to study what they were doing out in the ocean was observing the animals at the end of their time on land. They're fasting for up to a couple of months at a time, not eating or drinking anything. And then when they come back, you have to look quickly here, they come back nice and fat. So they're gaining a tremendous amount of resources. I'll flip back and forth between these a couple of times so you can see the skinny seal versus a fat seal. And in short, they're basically doubling and halving their mass on an annual basis. This is for the adult females. So that's incredibly physiologically taxing, but it is a very successful strategy for them because they're going to areas to feed that most animals cannot get to. So these reliable prey resources. So whenever I think about an elephant seal going out to sea to forage, I like to think about what it's like from their perspective. So I like to think of the middle of the Northeast Pacific. They're going out there thousands of kilometers, and this is what it's like out there. There's no cues of any kind, just the surface of the ocean and then the depths of the ocean. They're very, very far away from land. And actually, they're underwater most of the time. So the time at the surface like this is only 2 minutes at a time. And for about 23 minutes on average, they're underwater. So this is their world for most of their life, is hundreds of meters down in the Northeast Pacific. Just a very difficult thing to study because we cannot get there very easily. It's almost impossible to follow these guys. On a single dive, they can traverse more than a mile horizontally, and in some cases, up to a mile deep. So very difficult to follow them. So we need to rely upon technology to do that. So we can attach a whole variety of biologging instruments to them to sense information about the animal itself, its physiology, or its behavior, or the ambient environment. So lots of different biologging tags exist that tell us about those features. And so we'll go through just a couple of them. This is one of the more advanced ones that we use. It is a conductivity temperature depth tag. In addition, it is a satellite tag. So this one tag alone can give us position information. So this long antenna sticking up the front here is an Argos antenna that transmits to orbiting satellites to give us a latitude, longitude position several times per day. There's this little sensor up front here that measures temperature super accurately. This whole black contraption on top is measuring conductivity of the ocean. If we have conductivity and temperature, we can calculate salinity, so we can calculate information about water masses. Behind this, that you can't see, is a depth sensor. We have a pair of little metal pieces here that act as a saltwater switch to tell the tag when it's at the surface or when it's at depth. And then lots of circuitry inside, obviously, and then a big battery on the back to power it for months and months on end. Another type of tag that we put out is a GPS tag. So this one has a little GPS antenna up top and gives us really fine resolution tracking data. We also put out tags that can record things like the stroking pattern of the rear flippers. We can record the jaw motion of the seal to record feeding events. We can put cameras out on the seals to record their prey capture attempts and what they're eating. So all sorts of amazing things, and researchers and engineers are coming up with all sorts of new technologies all the time. And what's very exciting is that elephant seals are often the place where researchers use this technology first, in part because they're super reliable. They have that high site fidelity that I was talking about. So if we put out an instrument on one of these seals, it is very likely to come back with that instrument, and we can download the data and learn from that. And then the researchers that develop that can go and do something more risky and attach it to an animal that has a less likelihood of coming back. Okay, so what is this, some of the things we've learned from these tags? So the first thing, I'll talk about the horizontal movements of the animals. So if we were to zoom back in time to the early 1990s, and you were to ask a researcher, where do elephant seals go? This is the map that they would show you. This was the state of the art back then based on visual surveys. So where were the elephant seals? Well, they were where we were looking for them. There were a bunch of coastal surveys done, and we would occasionally see elephant seals. And so we thought that they were largely coastal. And then Bernie LaBeouf attached satellite tags to the seals in the late 90s, and they were shocked to see exactly how far the animals were going. These males and females were traveling halfway across the Pacific Basin and back twice per year. So the yellow lines there represent the adult females, and the the red lines represent the adult males. So you can see that both sexes are going just extraordinary distances twice per year. And then since then, we've been able to attach instruments to animals pretty much every year since about 2004, and to keep this long-term data set going. And this is now quite out of date. We probably have twice as many seals as instrumented as we have indicated here. But this gives you an idea about the density of data that we have now. So the animals are basically covering the entire Northeast Pacific. These are all females in this image. They cover all the areas along the Alaska coastline. They're going 3/4 of the way to Japan. We have animals that we've instrumented down in Mexico and the Channel Islands, and you can see that those largely go to some of the same areas. So we have a very good understanding about where these animals go. And that alone is incredible. But what's neat about instrumenting lots of animals is that we can begin to look at individuals and see some interesting patterns there. So in the next slide here, I'll show you just a few individuals that were very interesting for me during my PhD dissertation that I took a closer look at. So each number here corresponds to one animal, one adult female elephant seal, and each of them are doing some interesting things. Now, when I think about going out into the middle of the open ocean and traversing thousands of kilometers over months on end, I picture myself doing that and getting lost after the first day. Like it would be very difficult for me to orient and navigate in that environment. Yet these elephant seals can do that very efficiently. And they have some pretty unique abilities that enable them to do that. So animal number one here, it starts off by uh, going up here, pretty much in a remarkably straight line up to Alaska. It hits the continental shelf, spends some time foraging up there, goes back to the original place where it first hit that continental shelf, and then comes back along its original track. That's not exactly unexpected, but it's still pretty impressive that it can find its exact track. And then it decided to deviate into the Alaska Gyre region, spend some time foraging out there, and then it didn't have to go back to its original trajectory. It knew when it wanted to head home from the Alaska Gyre exactly which direction to head in. So that to me tells me that they have a compass sense. So these elephant seals know the direction of home at all times. Pretty amazing. Animal number two here was a fun one because we instrumented this animal on three separate occasions. So 3 years, all during the post-breeding migration. And so each color represents one trip, so across 3 different years. So you can see that this animal was very, very consistent. So each year of these 3 years, it went out to almost the same spot and came back along very similar trajectories. And all 6 times the animal passed this location, it went over that one spot. So I don't know, there might be something interesting going on there. I'm not sure what. And then animal number three here, one of our Mexican animals that I tagged down in the San Benito Islands, just went out and back, but over a very, very straight trajectory. And again, that's very challenging to do because there are different currents in these areas, and just knowing exactly which direction to head home is very challenging without visual cues of any kind. So in short, elephant seals are just amazing long distance navigators. I'm pretty sure they're the most accurate animal navigators on the planet over long distances. We're hoping to publish a paper pretty soon to detail that. But it's pretty amazing what they're capable of. Professor Beltran took some of this knowledge and decided it'd be very interesting to kind of prove that the animals have a map sense so that they have a compass and they know how long it takes, basically how far away they are from their home colony and the direction they need to go in. So in this next image here, the first part of her study was to take all of our elephant seal tracks and identify the point where the animals turn around to come back home. And that sounds like a weird thing to do, but if you know when they're turning around to come back home at the end of their trip, then we can start to study some interesting patterns, like how long it takes them to come home and how quickly they travel. So what she did was for each of those yellow dots right there, for each of those animals, she noted the date that the animals were turning around and the distance. And so it turns out that animals that are farther away turn around earlier in the year. And that sounds very intuitive, right? I mean, if I'm farther away from my destination, I need to leave earlier to get there. But that's actually a surprising result because that implies that the elephant seals at any given moment know exactly how far away from the colony they are. And then we've already shown that they can orient very successfully with their compass sense. So between those two facts, we can basically show that the animals have a map sense. They know how far away they are from the destination and they know how to get back. So that's a pretty neat thing to be able to document, especially in in a marine mammal. This is hard to document even in terrestrial animals. Okay, so this next image here is the tracking data animation. So this is basically a bunch of animals all during 1 year. So this is the short post-breed migration. The females on right now, they come back for the annual molt, stick around the land for about a month, and then go off to see a long post-molt migration. This is the important one because they're growing a fetus inside of them, and then they're getting as big as possible for the breeding season, coming back all about the same time in mid-January to give birth to a single pup, nursing that pup for about a month, and then they're off again on their post-breeding migration. So the big question is, what are they doing out there? Like, why do they need to travel so far out into the ocean to be successful? I always like to compare elephant seals to California sea lions because they both exist along the coast here, but yet do very, very different things when they're out in the ocean. You guys are probably aware that California sea lions are largely coastal. They stay pretty much on the continental shelf and just go up and down the coast, whereas the elephant seals are traveling thousands of kilometers out into the open ocean. So I'm not an oceanographer myself, but this is a very stylized diagram of what's going on out in the middle of the Northeast Pacific. We have this gyre boundary that separates the subarctic gyre, which loops around, and then the subtropical gyre, which loops around to the south there. And this is, as I mentioned, it's just a stylized diagram, but what we can do is take actual measurements using satellites of the ocean and identify where this gyre boundary is in real time using satellite altimetry data. And then we can also lay over that where the seals are actually going. So this is a stylized diagram. Now I'm going to show you some real data. So this black line is that gyre boundary using the altimetry data. And then the heat map here is showing where the elephant seals are spending most of their time. So you see we have some elephant seals going all over the place, but where they're actually spending most of their time is very well aligned with this gyre boundary. So we think that's a very important productive area that's very reliable for them and that not many other predators can reach. So this is just the bounty for them. This is the grocery store where they can just walk in and get food every single dive. And that's a pretty amazing resource. And again, to compare that with California sea lions, there can be boom and bust years with the sardines and anchovies, for example. It might be a great year for feeding, or it might be a really bad year. And we see that reflected in their reproductive behavior. So there are some years when many pups of California sea lions die of starvation. And there are years when the pups do great and they expand the population. And with elephant seals, they just kind of do well every year. It's very reliable out there. So that's kind of a fun thing to learn. Okay, so now we can go into a little bit more detail about their diving behavior. So we know generally why they're going out there to go to this gyre boundary. So we can take a more detailed look at their diving behavior and see that this is a trace from one of those time depth recorders. And you can see they're routinely going down to anywhere between 450 and 650 meters, occasionally much deeper than that. And we can also lay over that some data collected from jaw acceleration loggers. So each of these red dots corresponds to a jaw acceleration event. And then we can know exactly when the animals are feeding, which is pretty amazing. With most marine mammals, this is a very hard thing to study because it takes us attaching a tag to the lower jaw of the animal, which is kind of tricky, but then also having the animal come back and allowing us to collect that instrument back off the seal. So pretty challenging to do that with most species, but we can do that with elephant seals and therefore learn a lot about what's going out there in the middle of the Northeast Pacific. And then in addition to that, we can attach cameras to the seals, and our Japanese collaborators, and I'll talk a little bit more about them later, have collected some amazing data from our seals that we've equipped with satellite tags and time depth recorders and jaw acceleration loggers. And we can also attach a camera to them to study exactly what they're seeing and what's going into their mouth right before they feed on it. So these were some of the first images that we got back from those tags. And this is a myctophid fish, and this is, it's a lanternfish. And on the right here, we have a bathylaggid, which is a deep sea smelt. So in short, we learned that probably they're eating mostly fish out there. And that was just the start of a whole series of studies to learn more about their diet, because just really not much is known about that deep ocean area between 200 meters and 1,000 meters that some people call the twilight zone, is how dark it is. Okay, so these were just some still images, but we can also get video, which is pretty exciting. So we'll play that in the next slide here. So this again is from our Japanese collaborators, and this is video from the head of elephant seal, hundreds of meters down below the surface. And it really takes some collaboration with fish experts to identify some of these fish species. I would not be able to do this myself. But just little glimpses of fish right before they're eaten are enough to identify them, usually down to genus level. And mostly they're pretty small fish, but occasionally they go after larger fish. They do go after squid. We've learned that they're very opportunistic feeders. So in the next slide here, you'll see a whole variety of different species, both fish and squid, that they go after. It is a fish-dominated diet, but like I said, we do see lots of other diet items. And what's really fun is that we actually had two different projects going on simultaneously that was looking at their diet. We had our Japanese collaborators putting out these camera and video loggers. And then there was a graduate student in Dan Costa's lab who was looking at the fatty acid signatures of the animals. So this is using the you are what you eat phenomenon. So if you take a super tiny little blubber sample from the seals and then analyze the fatty acids that are in that, and then compare that to potential prey items, you can get an idea about what they're eating. And these methods aligned very well. So the fatty acid analysis is a great way to look at just generally what they're eating. And then the cameras are a great way to identify individual species because we can more clearly identify them in photos than with the fatty acid method. So yeah, it's always nice when research methods agree like that, research findings agree like that. Okay, so then one other really fun thing about some of these studies looking at their feeding behavior, we began to ask questions like, okay, that's great, we know what they're eating, but how are they finding all of this prey down there? They're literally feeding on up to 20 or 30 little fish per dive, and they're doing 20 to 30 dives per day when they're out in some of these areas. To be that efficient at foraging, they need to be really good at finding these prey items. Yet it is completely pitch black dark in these environments. Even on a bright sunny day, it is pitch black at 600 meters below the surface. So how are they finding these prey items? So we had two different ideas. One was basically very sensitive eyes. So even though not much surface light is reaching those depths, many of their prey items are bioluminescent. So they might be using bioluminescence to literally see the prey that are emitting light and go after them. And then the other hypothesis was using their extremely sensitive whisker bed. So if anyone's seen a close-up picture of an elephant seal, they have this amazing whisker bed with whiskers that project forward, and they're very firm whiskers. So they have very special structures. So it was thought they might use those whiskers to find little turbulent structures in the water after a fish or squid swims by, and they're able to locate the animals, the prey items based on that. So this plot on the right here shows the depth that many of their prey species are located, so between 400 and 650 meters. And then on the right here, you can see the percentage of time when the whiskers are protracted as a function of depth. And you can see that it actually matches pretty well. So the whisker protractions co-occur with the presence of their predominant prey species. So that was a great little piece of information. But then we needed more information about this bioluminescence detection. Are they actually just seeing the prey? So our collaborators put out very, very sensitive light measuring tags. So basically, these little tiny bits of light emitted by these prey species at depth can be detected by this very sensitive tag. And in this plot here, we have the diving pattern of the animal. This is just one dive. So diving down to nearly 500 meters, spending some time down there, and then coming back up to the surface. The black squares correspond to feeding events identified from the jaw acceleration logger. And then the bottom plot here shows irradiance, or basically the light level detected by the tag that is specifically designed to measure bioluminescence. And you can see that here only one of these feeding signals corresponds with that irradiance level. So that's interesting. So there's lots of feeding going on without bioluminescence. So this was an interesting discovery, but it took a lot of instrumentation and collecting more data to kind of refine this result to see if it was a general pattern. So bear with me here. This graph is a little bit complicated, but basically just this one on the left here is the percentage of bioluminescence with feeding signals. So basically, if there's a feeding signal, how likely is there to be bioluminescence along with that? And then also with bioluminescence, how likely is a feeding event to occur? So with this one, if bioluminescence was detected, almost all of those, like 90% of them, had a feeding event. So if a seal sees a prey item that's bioluminescing, almost always it captures that prey item. So that's pretty cool. And then on the right here, it's kind of the opposite. It's basically looking at, of all of the prey captures that we observed, what proportion of them were associated with bioluminescence. So it turns out only up to 20%, but the deeper you go, it kind of trails off here. So there's an average of maybe like 10% of the feeding events had bioluminescence. So that means that if a seal sees a prey item, that's a dead giveaway. They can go and capture it very easily. But that's only a small fraction of the prey items. So we think that 90% of the prey items that the animals are feeding on are detected from the whiskers, which is pretty amazing. They're just feeling their way down there, feeling those turbulent structures in the water. Okay, so let's do a little deep dive into the diving behavior of the seals. So this is a very sped up animation of an elephant seal diving record. Each of these dives lasts an average of about 23 minutes, and they spend about 2 minutes at the surface. So it's obviously very sped up, but they do this continuously when they're out at sea. So day and night, 24/7, for months on end, they are diving. And I find it a little bit difficult to grasp the depths that these animals are going to. So I find it helpful to put it in the context of some of the world's tallest buildings. So here's whatever 10 or 12 tallest buildings in the world. So pick your favorite building and let's compare that to an elephant seal dive. The deepest dive we've been able to record on a adult female elephant seal, 1,750 meters. So this is just wild. I mean, this is like walking up the stairs, up the world's tallest building, up to the tippy top, having lunch up there because they're feeding on these dives, and then walking all the way back down, all on one breath, which is just absolutely wild to think about. And to top it off, if you were doing that, if you were to hold your breath for that long, you would want to take in a deep breath, right? Before you hold your breath for that long, so you can maximize the amount of oxygen you have. The elephant seals actually exhale before they dive. So these are pretty amazing animals, and they're able to do this continuously. Okay, so how does that compare to some other species? This was a nice review paper that was written a few years ago. And this is the summary figure from that. And this is pretty complicated, but I just wanted to show you how amazing elephant seals are relative to other marine mammals. So on the left here, we have the maximum dive depth of elephant seals versus other pinnipeds. And you can see that just they're just in a different rank altogether. So elephant seals are diving just far, far deeper than other pinnipeds. And really, it's in line with some of the deepest diving toothed whales, which is just wild because of their difference in body size. So the graph on the right here relates body mass on a log scale to maximum dive depth. And basically, you can see that in general, let's take all these gray bars, the toothed whales, you can see that as body size increases, the maximum dive depth increases. And that's an expectation that a lot of physiologists have because they have a lower metabolic rate and can store more oxygen. So you'd expect larger animals to be able to dive longer. What's really interesting, though, is that elephant seals are really standing out here because for their body size, they should only be able to dive to 400 meters, but they can dive like 4 times deeper than that. So they're just absolutely amazing at being able to do dives relative to their body size. And that's important because they're going after relatively small prey. So we think that that is the unique niche the elephant seal has found and why they're so successful. They can get down to these amazingly deep depths where only a few other species can dive to, and they're relatively small, so they can make a living on very small prey. So that's just a very unique niche. And so that enables them to have this very reliable prey resource that is not in competition with other species. Okay, so that's kind of a quick look at their diving patterns. Let's take a kind of a deeper look into the different shapes of the dives. So when Bernie LeBoeuf and his colleagues first got back some of the time depth recorder records, this is what the records look like. So there's just a series of dates, times, and depths here. And we can use some fancy computer programs to sort them out and determine the start and end of each dive, and then the shape of it and some interesting characteristics. And it turns out that there's one type of dive that was super interesting to a lot of people, including myself. I based a lot of my PhD dissertation on these sea dives or drift dives. So these images are just stylized diagrams. Let's take a look at some real dives here. So this is a kind of a work day for an elephant seal. Sort of funny, 9 to 5 p.m. The animal's diving continuously, as I mentioned. All of the dives during the first part here are foraging dives. They have lots of up and down wiggles chasing after prey at the bottom there. But then we see that there's this series of drift dives where they're just passively drifting during this phase at the mid part of the dive here. And it turns out that some of our collaborators put acceleration data loggers and gyroscopes on these animals, and they're not just kind of drifting a straight line down. They're doing this very strange behavior where they flip onto their back and just passively drift down in a spiral, which is pretty wild. More recent studies have been able to document that they're actually asleep during that phase of the dive. So these are sleep dives. So they dive down to about 200 meters, flip onto their back, passively spiral down for about 10 minutes, and fall through the water column several hundred meters, and then wake up and swim back up to the surface. And these little cat naps are how they get all of their sleep when they're out at sea. So they're not sleeping at the surface or hauling out. They're just getting little naps on these drift dives. So that's just inherently interesting, but a few folks discovered that there's some interesting things you can do with these data. And one of them is by zooming into one of these individual dives and looking at the rate at which they're drifting. So this is a real drift dive, and you can see that this has a particular slope to it. And sometimes we see slopes that are a little shallower than that. So that's an animal that is very skinny. It doesn't have very much fat. So fat is positively buoyant. So if you're skinny, you sink faster. And as you get fatter and fatter, eventually you'll actually float up instead of sinking down. So this nice fat seal here, will float up to the surface. And what's really neat is that we can measure the rate of change in this drift rate from day to day and figure out how much fat they're gaining, how much they're changing their buoyancy. So this is a plot of change in buoyancy as a function of the progression along the migration. So we're starting off in June, going all the way to January. And this is basically showing that whenever these dots are above that red line, the seal is in positive energy balance. It's putting on fat. So there's a nice little pulse here. It found a nice foraging patch and then went below, so actually lost some mass here, and then found another foraging patch, then went back to neutral, then found a really nice patch and stayed there for a while, and was in positive energy balance for a long time, and then was neutral for a while as she headed home, and then finally made it back to the colony. So this is just one individual animal, but it tells us a lot about the foraging success of this one animal. And this happened to be one that I tagged down in Mexico. So left Mexico here, headed up north, went out here, and then the bulk of the time the animal spent out in that one little area where it had a lot of success, and then headed back along that trajectory. So again, that's super interesting from a single animal's perspective. But what we can do is accumulate all of the data that we've collected. Here are a whole bunch of adult females, and then run that same analysis and figure out where they were actually having their foraging success. And that lights up the ocean like this. And you can see that matches very well with that gyre boundary that I was mentioning earlier. So it turns out that the elephant seals are spending lots of time in that area and they're successfully foraging in that area. And what's neat is that we can look at some of these other areas too. So like along the Aleutian Islands here and along the continental shelf break up near Alaska, the animal is also having lots of foraging success there. So it's just a very powerful way for us to see on a day-to-day basis where the animals are feeding successfully. And I always like to point out to folks that this is incredibly useful information because you can not really get this for any other marine mammal species, especially on this day-to-day basis. It is very difficult to collect this type of data on a species. So to be able to do this, especially over this spatial scale and this number of animals, allows us to address some super interesting questions. And so there's been lots of follow-up studies that have looked at this data set and answered questions like, okay, how does this relate to their sleep? And how does this relate to their previous history and who their mom was? Does that relate to their future foraging success? So lots of amazing aspects there. So it would be impossible for me to talk about all of the different studies that go on, and this would be like a whole like quarter long class at the undergraduate level. So to pack it all into 1 hour here is pretty challenging. But I'll just mention a couple of other quick things. a lot of other folks around the world find the elephant seals and the research that we do pretty interesting. So we often have national and international media coverage with the results. So everything from that at sea behavior to how the animals can be used as predictors of deep ocean productivity. Roxanne Beltran just published a study in Science magazine, which is very highly regarded in our field, about using elephant seals as indicators of the deep ocean. There's been some great work done on the acoustics of the seals and how they can remember their calls across years to save time and energy battling each other. So just, yeah, lots of amazing stuff. And I think this has inspired a lot of research around the world, which is pretty exciting. And I mentioned earlier on that we try to do the best job we can with engaging as many undergraduates and early career scientists as possible. And this is just a kind of a class photo of the class that I co-teach with some folks, um, called the Large Marine Vertebrates Field Class. And because of this proximity with the colony de Año Nuevo to our university, we can literally take these students out in small groups each morning during the breeding and molting season, and they get hands-on experience and do some amazing work. And we try very hard to have a diverse group of students participate because traditionally, in science in general, but especially marine mammal science, has been very uh, biased towards really white males. So we try to incorporate the diversity of the student body in the class. And this really has launched a whole variety of careers because they can walk away from that undergraduate degree with some really unique skill sets, immediately get jobs in their field and set them off onto graduate school and professorships and other types of jobs. So yeah, we hope to continue that long into the future. Okay, so I see I'm running a little bit short on time, so I'll just pause there for questions. But before you guys ask questions, I just wanted to make a quick plug to the funding agencies that have funded Professors Beltran and Costa for the past couple of decades. As I mentioned before, this is a very difficult thing to keep going for such a long period of time. So it requires principal investigators that are continuously coming up with good ideas and writing good grant proposals and funding graduate students and postdocs and researchers to go out and do all the work and write papers. So yeah, so the Office of Naval Research, the National Science Foundation, Packard Foundation, Beckman Foundation, and a whole variety of other sources, including some private donations, have enabled this to happen. And just one quick sort of bummer is that Professor Beltran just recently got some of her Office of Naval Research money that was granted to her pulled back. I'm sure you guys have heard about the National Science Foundation, NIH, and different federal agencies canceling grants. And unfortunately, that has happened for our elephant seal program. So it's a little bit of a bummer, but fingers crossed that the other grants will not be canceled and we'll be able to continue the work into the future. So on that lovely note, I'd be happy to answer any questions you have. And yeah, thanks for your attention.
Well, thank you so very much. That was absolutely thrilling.
Yeah, and I just realized that it got
Very,
dark, so I'm going to turn on the light in the room here.
very edifying. Yes. So let's see what's a cooking in okay, so Sam is saying hi.
Hi,
Um,
Sam.
um, Sandra's wondering how to connect with you. Um, it says Lenny is saying something about the video of the elephant seal with a device attached to it. So where is the actual camera? The
Oh,
device,
yeah. So, yeah, that
yeah.
was, I think, in reference to the. I should actually just play that while we're doing the Q&A session here.
If you
Um,
don't talk during the video, it's better. If
oh,
you
okay.
if you're showing the video, talk after because
Okay.
then your voice doesn't break up.
Okay.
If you don't
Um,
mind.
I'll just play a couple of seconds of it then, just so you guys can see the video for a second. Let me share my screen again here. Okay, so hopefully you can see that. So yeah, on that particular seal, that was actually a satellite tag on the head that you're seeing, and the camera was actually attached to the kind of the shoulder or back region of the animal. And so that's not a normal thing that we do. That was just to test out that particular tag, but it was kind of neat because we could see the satellite tag. And so it's a good thing to show for talks like this. But normally we would attach the camera to the head so we could kind of see what the seal is seeing and see those prey items before they go into the mouth.
Mm. So most of the tags then, or maybe all tags, have to be placed on females because
Oh yeah, so
the
yeah,
males
so
won't
I
let you near them, right?
So I forgot to mention that. So we do use a chemical immobilization procedure. It's safer for the researchers and safer for the animals to do that. So with that approach, we can actually instrument any age class. So we've instrumented weanlings, yearlings, 2-year-olds, 3-year-olds, adult females, subadult males, and even adult males. So it is possible. I personally don't like chemically immobilizing adult males because they have some interesting physiology, like in their extradural vein, which is where we inject the immobilizing agent, it can actually, like the blood flow can actually reverse in there. And yeah, there's some weird stuff that happens with the adult males. So we basically just have one person, who's Dr. Dan Crocker, who we kind of trust to do that. So it's not a common thing that we do, but we have done that, a number of times in the past to get some of those tracking and diving data sets. And I'll say that the female seals are actually much more scary. Like those are the ones that will bite us. The males, as we're walking around the colony, they're big. I mean, they can seem scary, but really they don't want to expend any energy on us puny little humans. Like they only care about the other males that are around. So
Uh,
occasionally there'll be an aggressive male, but it's mostly the females that will sneak up to us and try to bite us. So the
so the
females we have to watch out
two-legged
for.
human beings are not no competition for them and not worth worrying about.
Yeah, what's funny is I actually, I probably shouldn't say this, but I sort of use the males as a blockade from the females. So it's safe to be near a big male. It's kind of like a big boulder.
That's fascinating. You do what you have to do. I
Yep.
mean, but the males, when they do fight, I mean, in order to gain ascendancy and alpha status in a harem, how far do they go with the fighting? They don't really kill each other. They go quite a ways in terms of hurting one another and bleeding,
Yeah,
right?
so that's so some of the acoustics work has been very, very interesting. So this is Dr. Carolyn Casey and Colleen Reichmuth out of UC Santa Cruz. And what they've done is some very detailed playback experiments that basically show that the seals are very, very good at not fighting when they don't need to. So if two different males have interacted with each other in the past, they each make a unique call. It sounds very similar to me, but they can tell each other apart just from those calls. And if they interact again, they'll both call and they'll remember the outcome of that previous interaction and behave accordingly without needing to engage in physical combat again. So, these two males like might just call and they're like, oh crap, like you beat me up last week, so I'll just back away and that'll save any physical altercation. So most of the interactions we have between males are just like a head lift or a call, and that's it, and that's the end of it.
Really?
It's when two males have not interacted previously and they both think that they can win, then they engage in that physical combat. And most of the time, that usually ends pretty quickly. So just, a couple little, jabs to the chest shield, and then one of them realizes, crap, I'm outmatched here, and then they back off on rare occasion, like it doesn't happen a huge number of times per season, but sometimes, especially during the late breeding season when most of the mating happens, um, two males will think that they're equally matched, and those will last a very long time potentially. I've seen a fight last more than a half an hour, and they can get very bloody, and they do have very thick tissue on their chest shield, so they actually tend not to get severely injured. There can be some lacerations, but I've only seen one male die from a fight, and it was basically a canine to the top of the head, and it broke the skull. And it was a very mismatched fight. So it was a adult male going after a subadult male, and I think that maybe the subadult male just didn't see him coming. So they can normally defend themselves by facing the male that they're fighting with. So it's a very extraordinarily rare thing for a male to sustain severe injuries or die during a fight.
Mm. Well, best to keep alive, I suppose, and stay alive. Yeah.
Yep.
Uh, okay. All right, let's check what else is here. Um,
I'm seeing one question about what is the difference between a reserve and a preserve? Yeah, there's lots of different names for these different areas. Um,
go to
so
it.
the reserve, I call it that because it is part of the University of California Natural Reserve System. So it is a university reserve. So it's kind of dedicated to research and teaching activities. It is also a preserve out there. So that's a distinction the state park gives the area that's out near the elephant seal colony. And so it's called a natural preserve. And that enables it to be basically a permit only entry. So even members of the public, when you go out, technically you are getting a permit to go out there. So if you come out during the breeding season and go out on a guided walk, that is a permit. You're getting a permit to go out on there. And then during the off season, which is starting now and throughout the summer, you have to check in at the Marine Education Center and get a map, and they give you some basic instructions, and that map is your permit. So it's basically a way for them to control the access out there. Otherwise, people would just be allowed to go out onto the beach and disturb animals and do bad things out there. So it's both a reserve and a preserve.
One for research and one for protection in terms of the number of people who go out.
Yep, exactly. Looks like Sarah
Sand.
was asking, yeah, how do they deal with the sand that gets in their eyes? Um, so the short answer is they have amazing tears and they can just like blink once or twice and eliminate all the sand out of their eyes. Like I can't do that, like even if I put lots of water in my eyes. So they have an amazing ability to do that. And I think that would be a great future study because I think a lot of people and other animals could benefit from that. But I have no idea what is special about their tears and their anatomy that allows them to do that. But yeah, I've seen even a newborn pup, like minutes old, the mom flips sand, covers the pup's eyeballs with sand, the pup blinks once and there's no more sand in its eyes. It is absolutely amazing. So I'm not sure we know exactly how that's done, but it'd be a great study.
The mother does what? I'm sorry?
Oh,
She
flip sand. So oftentimes the seals, when they are overheated or disturbed for any reason, they flip sand using their foreflippers onto their back. But when they do that, sometimes the sand lands on the pup instead of them, and if it lands in their face, they can just get an eye full of sand. It's one of the many things that are hard to watch out there.
Oh, heavens. Okay. But okay, let's see. Bing is asking about use of the camera light. Does the camera actually have a light or not?
Yeah, so it's a good question. Our Japanese collaborators, when they first brought us the camera that they wanted to put out on a seal, so it's like this first prototype, and it was actually my job to work with them to attach it to the seal. And they showed me how it worked and how they wanted to attach it, and it all looked good. But then I realized, yeah, like one tube was the camera, and then the other tube was a flash, like a camera flash. And I got to thinking to myself, if the seals are down deep and their pupils are wide open and they're going after prey items, at this time we didn't know how important the bioluminescence was relative to the whiskers. That might prevent them from feeding if they're just blinded by this flash all the time. And so I convinced them to put a small filter in the flash such that it would only emit near infrared light. And so that's why a lot of the images that you see of elephant seal prey items are purple because it's really not a full spectrum light. It's just that very narrow wavelength that is just outside the visual range of the seals. So yeah, I had the same concern, and we addressed that with a little filter, and then all of the future cameras and video loggers have had that same filter. So yeah,
How's your Japanese?
not good.
Okay.
Fortunately, they speak English.
Okay, so here, presumption, is there a presumption that sleep dies as opposed to something? Oop, lost him. Let's see. Sleep dives for protection from predators rather than sleeping at surface or close to the surface. Something to do with predation. if they're going to sleep higher up?
Yeah, so one of Professor Beltran's recent studies a few years ago, she calls it the landscape of fear. So it's sort of a strange term, but if you're an elephant seal, you need to worry about finding enough prey, but also you need to worry about not getting eaten. And so the kind of landscape where you need to worry about getting eaten is that landscape of fear. And so we think that the seals have adopted this strategy of sleeping during these drift dives in part because that is deeper than the depths of their predators. So if they can sleep during that period of time, then they can be safe and not have to worry about a predator attacking them. If they were to sleep at the surface, even out in the middle of the Northeast Pacific, that could put them at risk because many of their predators are visual predators that usually tap from below. So being deep in nice, deep, dark waters and sinking down is a nice strategy for them. And interestingly, she was able to show as well in this paper that the seals do something. So you can also do these drift dives and sleep during daylight hours or during nighttime hours. And it turns out that elephant seals are better foragers. They can more easily capture prey during the nighttime because the prey are a little bit shallower in the water column. And
Mm.
then during the day, they go a little bit deeper. And so it takes them longer and they get fewer prey resources. So if you're a hungry elephant seal that's just been fasting for a month or two on land, then there's this trade-off. Do you sleep during the day or during the night? If you sleep during the day, then that's a little bit more risky. If you sleep at night, then you could potentially be preventing some foraging opportunities. And so it turns out that when the animals are very skinny, they do the more risky thing of sleeping during the day. And then as they get fatter, they switch to sleeping more during the nighttime hours. So we think that they're being very adaptive and like understanding their own needs and assessing risks and everything. So it's a complicated life for them out there.
Mm. Okay.
So the risk from predators, is that like great white sharks or orcas? Or...
Yeah, so it's a super hard thing to study because if a seal gets eaten by a predator, then we don't get our data back. So, it's really hard to know what's going on out there. But we do see animals coming back with what we think are shark bite wounds. And there has been evidence of people going out and seeing like an elephant seal that's been ripped apart that, I think only an orca could do that. So we have these little bits of anecdotal evidence, but it's just a really hard thing to study. Professor Beltran is looking at one particular aspect of that question, and she instrumented lots of juvenile seals with this special type of tag that has an acceleration data logger in it that transmits the acceleration data. And so with that, she can she studied juvenile seals, which have a higher mortality rate out at sea. So she puts out a bunch of these tags, and then she hopes that all of them survive, but inevitably some of them are preyed on by orcas and white sharks. And so those that die out at sea, she can go back and look at the transmitted data and see, was that animal feeding for the week prior to that? So then she can determine if the animal starved or if it was a predation event. And so she has graduate students right now that are addressing those questions. And so I don't know the answer. Like wait a year and her graduate students will publish those papers. But she's addressing the starvation versus predation question. But in terms of like the proportion of white shark versus orca predation or maybe some other predator, yeah, it's just really hard to study. And that's going to require some like new technology that we don't have yet.
So the technology that you folks use and keeps coming, is improved or comes up with other strategies that may help you more. Who actually does it? Is it on staff that you have these engineers who are actually building and building these units that you attach? Or is it some other facility somewhere that has these engineers and researchers and designers who can come up with this? And how do you attach them? I mean, with what?
Yeah, so who comes up with all these designs of new tags? It is not us. So at UC Santa Cruz, we're really good at animal handling and ecology research and things like that, and not so much of the engineering side. So we rely upon collaborators and commercial businesses to do that work. So the satellite tags that we use and time depth recorders are made by one of two commercial companies. So there's a commercial company up in Washington called Wildlife Computers, and they just make these tags for lots of different species, and they've been making them for elephant seals for years. They're kind of expensive, but they are very reliable. So that's one of the major expenditures in these grants that the professors get. There's also a company over in St. Andrews, Scotland, called the Sea Mammal Research Unit, and that CTD tag that I showed an image of is an example of that tag. And again, they're very expensive, but they've spent years, in research and development and production making those tags, and that's been great. We can also work with folks like our Japanese collaborators. So Yasuhiko Naito, Akinori Takahashi, and Taiki Adachi work with the company in Tokyo called Little Leonardo. And they're the ones that, it's a bunch of old Japanese guys who are in their previous life were medical device manufacturers for humans. And they decided to start their own little business to study animals, which is great. And so they custom fabricate the aluminum tubes and all the circuitry and programming and everything that goes on to have these acceleration data loggers and cameras and still image loggers. So so for those guys, they're doing all their in-house engineering through that Little Leonardo company. And then we work with some other manufacturers like the Cornell Lab of Ornithology. They have the infrastructure to develop some of these types of tags. And so one of Professor Beltran's students is working with Holger Klink, which is a researcher working at the Cornell Lab. And he's just working with his local engineers to develop a passive acoustic recorder, and she's attaching those to animals. So yeah, it's kind of a mix of commercial companies and university collaborators. And then how do we attach these things? So we do just epoxy the tags to the seals. It sounds very simplistic, but we have some very detailed methodology for doing that. In short, we wrap the tags usually in electrical splicing tape. That basically just attaches that really well to it attaches well to the tag. I should mention before I go into detail about the attachment that our goal is to attach it very firmly to the seal. It needs to be able to last months and months out in the open ocean on the back of a seal. But then we also want to be able to detach it from the seal when it comes back. So it needs to be attached really well, but easily removable. So that's kind of a hard balance to reach there. So the first thing we do is we wrap the tag in electrical splicing tape and then use cable ties, just like normal zip ties you get at Home Depot, and attach the tag to a piece of mesh netting, just nylon mesh. And then so the only thing that's attaching to the animal is actually that nylon mesh. And then we epoxy that to the seal's fur. So we just mix up some two-part epoxy, puts them on the fur, puts them on the tag, put it on. The seal goes off to sea, it comes back, and then all we have to do is cut those cable ties and the tag pops off. So the electrical splicing tape basically prevents the tag from being chemically glued to the seal. It's just being held on by those cable ties. So when we cut the cable ties, the tag pops off. That epoxy patch will remain on the seal until its next annual molt, and then it just pops off.
I was wondering about the molting because the molting they'll lose everything with the molts.
Yep. So if we don't recover the instruments in time, then they will just lose them on their own, and then we have to go and find them in the sand. So we like to recover them before they molt it off. Otherwise, we might lose the instruments.
Okay. All right. Let's see. Have you still got time?
Yeah, got as much time as you want.
Okay. Here we go. Let's check. So I'll answer. Oh, Ellie, this lecture is recording. And it has been recording. So you will find it online at the website, Friends of San Pedro Valley Park, in a couple of days. It will be uploaded to our website, and there you will be able to see it at your leisure. Okay? Now, Bing says something about gyre boundary comparing to upwelling. Well, you can read it. Let's say, how does the gyre boundary compare to upwelling zones in terms of food availability? There you go.
Great question. So this is a neat one because why don't the seals just go into the California current, which we know is super productive? In short, I think the California current is far more productive, but more variable through time. So if you're an elephant seal, over evolutionary time, it is actually advantageous, I think, to feed in an area that is overall less productive but more reliable. So yeah, the California current, like definitely very strong upwelling. There are just crazy blooms of amazing productivity, but also periods of time when there's no productivity. And out in that gyre boundary area, that is just, it's very, very consistent out there. And so even in a bad year, like we've had El Nino events, La Nina, we've had Pacific decadal oscillation cycles, there's been marine heat waves, all sorts of crazy stuff happening out there. And the seals tend to do okay in all these different conditions. Like we notice small changes, like they might have slightly lower foraging success or slightly lower pupping success, or spend a little bit more time traveling between foraging patches, but we just don't see the sort of negative effects that we see with California sea lions, which rely upon the California current ecosystem, where we have these boom and bust years. So yeah, it's kind of like I mean, both are obviously viable strategies, feeding the California current and feeding in the gyre boundary area and the transition zone. And I think just there's disadvantages and advantages for either one. But if you were to ask me, like, which would I choose? I would choose to go out to the gyre boundary because it's a little bit safer out there, fewer predators, and it's a very reliable prey resource. So as long as I have the physiological capability to get down there, which not very many animals do, then I might as well take advantage of that very reliable prey resource.
Well, when you're mentioning this boundary, this line that you can project across the ocean, where they end up most of the time, or that they have a lot of resources. When we were listening to the Great White Shark lecture, Sal Jorgensen was saying the sharks have this cafe place too, where they seem to find a lot of food. And you find them along because now they attach all sorts of things to sharks. And there is this sort of boundary line for them as well. And it's the most amazing thing. There is something in the ocean somewhere that there is this area of good resources for certain animals, whether it's sharks or elephant seals or whatever, and they sort of know where to go and they stay in it.
Yeah, and it's worth noting that like if you were to somehow ask an elephant seal, like, do they know about that? Like, probably they don't. Like, it's probably not a conscious thing. They've been evolved to go in that direction, and so it's probably just instinctual.
Well,
But
yes,
yeah,
there's something, I don't know how to describe it. I mean, only not from an anthropological point of view, but basically they sense or they learn or it's somehow passed along genetically that they go to the places where the food is.
yeah,
Uh,
yeah. And studying
they hang
exactly how they know
out.
that is
It's
challenging.
like the
Yeah.
cafe area, man. It's food. So you keep going back, you know. Uh,
Yeah, and it's worth noting that the place where the elephant seals are going is much farther north than the White Shark Cafe. So fortunately for the seals, those are different areas.
Yes, I mean,
Yeah.
yeah, I mean, they're different, but the idea is that
Yeah.
they find food in those areas at certain depths or whatever, but they find food, reliable food.
Mhm.
And that's amazing. They know, they figured it out.
Yep.
So, okay. Well, we'll ask a few more and then we'll have pity on you and release you from answering questions. Physiologically, what's the advantage of exhaling before the dive? Or actually the coming back up?
Well, yeah. So before they go on a dive, they exhale. So they remove as much air as they can from their lungs. So yeah. So if you're an elephant seal, like you're obviously able to stay down a long period of time. You have lots of oxygen stores. Most of that oxygen is not in the lung. It's in the blood mostly and some in the muscle. So they have an amazing amount of blood, and within the blood they have lots of hemoglobin, and they have higher hematocrit levels, so like more red blood cells than we do, and they pack more hemoglobin in each red blood cell. So they're it's probably not the right way to say it, but it's like their blood is designed to hold lots of oxygen. And then their muscle has a very elevated level of myoglobin. So, they can pass a lot of oxygen onto their muscle and utilize that throughout the dive as well. And then they just basically don't need that lung oxygen. Now, why not utilize it? Like, why not just take a full breath? Well, first of all, that it impacts their buoyancy, so that could be bad, especially if they're positively buoyant. It would take more energy to get down. But what I think is going on is that if they take a nice lung full of air, then their lung collapses at a deeper depth. I didn't go into this, but basically every single dive an elephant seal goes on, their lungs collapse. And this is just like a wild thing. Like they reinflate them on every single dive. If that were to happen to us, like one dive and I'd be in the hospital with collapsed lungs and like it would be bad. So they're able to collapse and reinflate their lungs on every single dive. Okay, so why is that advantageous? Well, if they collapse their lung, then that limits the amount of nitrogen transfer from the lung into the bloodstream. So a lot of people ask, well, why don't elephant seals get the bends like humans do if you dive, super deep
Mhm.
period of time, super deep and for a long period of time? And for us, like we would get the bends because we're breathing on scuba, we'd be breathing pressurized air. So that would be basically like pumping nitrogen into our bodies. And then when that comes out of solution as we come up, then that gives us the bends, it produces bubbles in our bloodstream. The elephant seals, in theory, could have the same problem. They're not breathing pressurized air, but if they take a nice deep breath and then bring that down with them, then their lung becomes pressurized, and a little bit of nitrogen that's in their lung at high pressure can be pushed into their body. And if they do these dives, day after day after day after day, they can accumulate nitrogen in their body, and then eventually they could actually get bent. That nitrogen could come out of solution, sometime they come up to the surface. So I don't know this for sure, but like I think that that's why they do that. So they kind of limit the amount of nitrogen that's being transferred to their body to limit that possibility of getting bent.
Mhm. Okay. And then there's a few interesting there are animals treated differently in the reserve and the preserve than anywhere else, for instance,
So,
other than just keeping the public at bay, so to speak,
yeah, so in general, we have a very nice setup at Año Nuevo because we are able to like have lots of public access and lots of research access, but it's all very controlled. And in other areas, it's just a little bit less controlled. So occasionally, elephant seals will haul out at other areas, at other neighboring beaches, and it is illegal to walk up to an elephant seal and disturb it, but a lot of times people don't know that. And so we'll get interactions between people and seal or dogs and seals, which can be bad for a number of reasons. So the protection basically just enables us to have rangers and docents and researchers present to ensure everything goes smoothly and everyone's following the laws out there. Um, that's very important because it's a colony there. Like these other places, there might just be an individual animal or a few animals hauled out. And so if those animals are disturbed, it probably doesn't have any population level consequences. But if someone were to disturb the elephant seal colony, then that could potentially have some big population level problems. So, we put extra effort into protecting the colonies where lots of pups are born. But yeah, the combination of being the reserve and the preserve is what enables that to happen there.
right. And I'll ask if I may, one more question. There was someone who was mentioning the undergrad students in your program
Mhm.
and the fact that how is it that they would be allowed to walk the beach or whatever? Are they safe? Do they know what to do in terms of, I mean, I'm sure they're well trained, but this person is concerned about these undergrads being there, perhaps with maybe not enough knowledge, or are they safe enough? So
Yeah,
there is a concern there.
it's a great question, and I appreciate that a lot because I'm the person who like thinks about safety the most out there, I think. So in short, the undergrads are never alone out there. So they're always under direct supervision of a highly experienced person like myself or one of the professors or one of the graduate students who's been doing this for many years. So yeah, we don't just let the undergrads roam around out there. So always under supervision and always after weeks and weeks of training. So we have this whole like fairly formal training course where we teach the undergraduates about the biology of the animals, about their natural history, about the work that we do and the reasons why we do that, the permits that we have to do that, what we're allowed to do, what we're not allowed to do. how to stay safe out there in terms of the proximity to the animals, what their behavior is, what to do if they encounter an aggressive animal. So we cover all of that, and they don't even need to know that much information, but we give it to them just in case, because they're always under direct supervision by much more experienced people. And I always like to say, like, we've been doing this class now since 2015, so this is the 10th year of the class. And in addition, we bring out lots of other undergraduates, and we don't just bring out someone for the day. Like, we're bringing out people regularly so that they can be very well trained up and then get consistent experience out there and learn from that. Um, and we've never had any of the students in the class injured in any way. I've been bitten by a seal before. So I would like to say that, I put myself in danger before I put, an undergraduate in danger. So yeah, hopefully that alleviates any fear there.
Well, I think you deal much more close up since you are attaching all of these various technological devices. So you are literally hands-on
Yep.
where they are not yet and will not be for a good little while, if ever.
Yep.
But it's interesting to know that how well the training, how good the training is, and how even if you overfeed them with information, better to overfeed them with information than to not, obviously,
Yep.
to keep them
Exactly.
safe. Yeah.
Yeah.
So, well, it looks like we've gone through most of the questions and my husband's questions. And so thank you so much, Patrick. It's been a fascinating discussion because it's so different from what I learned when I was a docent at the academy. And I was walking the halls and so on, and the information has certainly changed. The knowledge on these sea elephant seals has changed quite considerably. So it was good to catch up.
Yeah, of course. And I forgot to mention one thing. Can I do a quick screen share to share one other bit of information?
Sure.
Great. So 1 second here. Okay, so the thing I forgot to mention was that the Beltran Lab website is a really fun place to visit because she has live tracking data of the seals available there. So if you go to beltranlab.ucsc.edu, then you get to this website. And then if you scroll down just a little ways, you can click on this link. And this brings you to the live tracking. So these are like literally adult female elephant seals that we're tracking. And you can see that like the green one is just starting to head back. The purple one here, the magenta one is like been heading back for a while. Yellow one is almost back. We have a couple that are, feeding along the coastline, and one stuck up off the coast of Canada who's not even headed back yet for the molting season. So everyone can go online and follow those seals in real time as those animals come back for the molting season.
But when they come back to molt, they do eat along the way back, surely.
Yes, probably not as much as when they're at their distal foraging areas, but yes, they do eat opportunistically along the way.
Well, that's good to know. Yeah, reassuring. All right. Well, thank you so much again on behalf of everyone who attended. I'm sure everyone, and you could read the comments, obviously. We're just thrilled with today's lecture, and I will be in touch in the next couple of days. So many thanks.
Of course, yeah. Thanks for all the great questions.
Right.
Take care,
And so,
everyone.
good, bye-bye. And folks, I'm going to be ending the webinar. We're going to say goodbye to Dr. Robinson, who was kind enough to be with us today. And so here it goes. We're ending. If I can find just a sec here.
Oh, there's an end button there.
Okay. No, but I have to stop recording. Right?
Yeah.